Hello everyone, this section is for inclusion body. After learning this section, you should be able to know the types of inclusions in cytoplasm and their functions. Inclusions are common in all cells. They are formed by the aggregation of substances that may be either organic or inorganic. Let's see the shapes of inclusions. Inclusions can take the form of granules, crystals, or globules. Some are amorphous. The locations of inclusions. Some inclusions lie free in the cytoplasm. Some inclusions are enclosed by a shell that is single-layered and may consist of proteins or of both proteins and phospholipids. Some inclusions are surrounded by imaginations of the plasma membrane. Inclusions are used for storage, for example, of carbon compounds, inorganic substances, and energy, all used to reduce osmotic pressure by tying up molecules in particulate form. The quantity of inclusions used for storage varies with the nutritional status of the cell. Firstly, let's see the storage inclusions. Cells have a wide variety of storage inclusions. Many are formed when one nutrient is in ready supply, but another nutrient is not. The most common storage inclusions are glycogen inclusions, polyhydrocolonate granules, PHA for short, sulfur globules, and polyphosphate granules. This picture shows PHB. It is an electron micrograph of Bacillus megatherium. PHB is a polymer of beta hydroxybutyrate molecules joined by ester bonds between the carboxyl and hydroxyl groups of adjacent molecules. PHB inclusions are around 0.2 to 0.7 micrometer in diameter. They are seen as empty holes by electron microscopy because PHB is hydrophobic and is dissolved by the solvent used to prepare specimens. Look at figure B. It is structure of a PHB granule. PHB is enclosed by a shell composed of several different proteins, including the PHB synthesizing enzyme, the red sphere, and the PHB degrading enzyme, the green sphere, and the yellow spheres represent the phospholipids that are also found in the shell. Much of the interest in PHB and other PHA granules is due to their industrial use in making biodegradable plastics. Polyphosphate granules and sulfur globules are inorganic inclusions, observed in many organisms. Polyphosphate granules store phosphate needed for synthesis of important cell constituents, such as nucleic acids. The sulfur globules, you can see it on the picture. They are formed by bacteria that use reduced sulfur-containing compounds as a source of electrons during their energy-conserving metabolic processes. microcompartment. Some bacterial inclusions are unique and serve functions other than simple storing substances for later use by the cell. These inclusions are called microcompartments, such as carboxylsome. Carboxysomes are present in many cyanobacteria and other carbon dioxide-fixing bacteria. You can see them on the picture. Their polyhedral coat is composed of about 6 different proteins and is about 100 nanometers in diameter. Enclosed by the shell is the enzyme carbonate anhydrase, which converts carbonic acid into carbon dioxide. There are also many other inclusions, such as gas values and magnetosomes. The gas vacuoles provides buoyancy to some aquatic bacteria, many of which are photosynthetic. Gas vacuoles are aggregates of enormous numbers of small, hollow, cylindrical structures called gas vesicles. There's a picture. This picture is a free structure preparation of Anamina philosophy. This shows gas vesicles and gas vacuoles. Clusters of the cylindrical vesicle form gas vacuoles. Both longitudinal and cross-sectional views of gas vesicles are indicated by arrows. Then let's say magnetosome. Magnetosomes are intracellular chains of magnetite particles. They are around 35 to 125 nanometers in diameter and enclosed within invaginations of the plasma membrane. You can see the magnetosome on the picture. Each ion particle is a tight magnet. Magnetosomes are very interesting. Magnetosomes are involved in bacterial movement. The northern hemisphere bacteria use their magnetosome chain to determine northward and downward directions, and swim down to nutrient-rich sediments or locate the optimum depths in freshwater and marine habitats. Magnetotactic bacteria in the southern hemisphere generally orient southward and downward with the same result. Okay, that's all for this section. Here is one list for you.